Hello, everyone. Welcome to today's live broadcast, Gene Expression Analysis Using 3' RNA Sequencing. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Lexogen. Lexogen is a transcriptomics and next-generation sequencing company focusing on development of products enabling complete transcriptome sequencing. For more information, please visit lexogen.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page and follow the process of obtaining your credits. I would now like to introduce today's speaker, Dr. Behnam Abasht, Assistant Professor at the University of Delaware. Behnam Abasht joined the University of Delaware's faculty as an assistant professor in 2011. He received his PhD in quantitative and molecular genetics in France from INRA Agrocampus, Rennes, in 2006. He then completed a postdoctoral fellowship in quantitative genetics at Iowa State University. Dr. Abasht was a research geneticist and genomics project leader at Purdue Farms from 2008 until his appointment at the University of Delaware. His area of research is integrative avian biology with emphasis on fine mapping and functional characterization of quantitative trait loci, QTL, in chickens. I will now turn it over to Dr. Abasht.
Okay, thank you, the moderator. My name is Behnam Abasht. Good morning, everyone. My presentation is about gene expression analysis using 3' RNA sequencing. First things I would like to discuss is about the sample size in RNA-seq studies. Most of the RNA-seq studies has sample size of 6 to 12 biological replicates. Typically, we compare, for example, affected samples affected by disease versus unaffected, or we compare group A versus group B. And usually, the sample size is not so big. It's like a 6 to 12 biological replicate. And the cost per sample is about $400 to $500. And we have done several RNA-seq studies in the past. These are some examples of a recent publication that my lab conducted recently. And this is the major actually driving force to us, how we can reduce the cost of RNA sequencing. So when we need large number of samples to be sequenced, for example, when we do population-level studies of gene expression, and those of you who are familiar in EQTL analysis, we need to have maybe 500, 600, sometimes thousands of samples. So the question was how we could do RNA-seq in large number of the samples, and whether we can reduce the cost to $50 per sample. So I would like firstly talk about like RNA-seq, a conventional RNA-seq method. So those of you who are familiar, you know that we first isolate mRNA, purify mRNA, and then after purifying mRNA, we synthesize cDNA using protocols which are provided by the companies. And these cDNA library are used to make small fragments of DNA. It is generally called cDNA libraries. So with cDNA libraries, we send them to sequencing. After sequencing, people receive a large amount of data, like small sequence reads, like an example of sequencing reads that you receive after sequencing this sample. So there's going to be 100 or 50 base pair, depends on the protocols that you use. You can generate a small sequencing reads. After that, after sequencing all these samples, you need to map them back to reference genome. Most species of interest, there are reference genome available. So we can map these sequence reads to the reference genome, and based on the number of the reads that map to each gene, you can quantify how much of that gene is expressed. So this is a typical RNA-seq experiment, which is used by us and most labs who are studying RNA sequencing. So in the following slides, I will compare RNA-seq conventional method with 3' RNA-seq. So in the bottom here, you see there is a gene model. We have on the left here, we have 5' UTR. UTR stands for untranslated region. And on the left-hand side, we have 5' UTR. On the right-hand side, we have 3' UTR. And in the middle, we have coding sequence. So this is a structure of gene in the gene model. So when we are talking about RNA-seq, in RNA-seq, we basically sequence all the transcripts produced from the gene. So full transcript. The goal is to sequence a transcript, and it is basically taking everything and sequencing the transcripts. But if you compare RNA-seq with 3' RNA-seq, so here you can see that in 3' RNA-seq, we don't sequence full transcript, but we focus on the right-hand side. You can see 3' UTR. We only sequence 100 base pair from 3' UTR, and we use this information to quantify gene expression. So if we count all these transcripts one by one, so we could quantify expression of the gene. So if the goal is to measure gene expression, we don't have to sequence full transcript as people do in RNA-seq analysis. So for quantification purpose, we only need to sequence a small part of each transcript, which with 3' RNA sequencing, we can do that. We can just confine sequencing to the end of the transcript. So this will, from here, it is obvious that how much saving will be, because we wouldn't use all the materials, sequencing materials to sequence full transcript. We would just sequence a small fragment of each transcript. So if you consider, for example, this one is, this figure shows distribution of transcript length. It is in chicken. Because I'm studying, my studies are on, most of it, we are studying chickens. So I took all genes from our latest, actually this is not the latest one, the latest one just published. This is GALGAL4, which is the version before the recent one. Recent one just updated a few months ago. So I took all the transcript and look at their length and distribution of the length. As you can see, some transcript is as big as 26 kb. So there is a transcript with big size, and average the transcript length is 2,000 base pair. So now you are comparing sequencing 2,000 base pair instead of sequencing 100 base pair instead of 2,000 base pair. So you can see how much saving will be if we can just focus on efforts on a small piece of the RNA. So this was basically it shows that how the difference between 3' and 3' RNA-seq and conventional RNA-seq method. Another benefit besides cost saving, There are other benefits of 3' RNA-seq analysis. One of them is to detect alternative polyadenylation. Polyadenylation, we know that from basic biology, every time in eukaryotes, RNA is transcribed to RNA is transcribed from gene. At earlier stage, they are pre-mRNA. They are called pre-mRNA, which stands for premature mRNA. So there are a couple of things happened before pre-mRNA became mature mRNA. One of them is basically cleaving the tail of the transcript. Here, as you can see here, there is a cleavage site. This site is determined based on this consensus sequence here. Wherever this consensus sequence is, about 11 to 30 base pair down, this transcript will be cleaved. And after cleavage, we're going to have shorter transcript. And you can see here, this part, the part was cleaved, and the transcript is shorter than the previous transcript. And after this, at final stage, a poly A tail, as you can see here, poly A tail is added to the transcript. So this is basically happens for all messenger RNAs that are transcribed from genes in eukaryotes. So we're gonna have first premature and then mature RNA, and there is quite amount of variation in terms of where the transcript will be cleaved. It could be in different sites, and this alternative cleavage produce a variation in the transcript length at 3' UTR region. So some transcript can be from the same gene. It could be a transcript with a longer UTR region. It could be a transcript with shorter 3' UTR region. So there's going to be variation in the transcript length based on the fact that where this cleavage will be happen. And this has implications because some of the RNA silencing mechanisms is related to, for example, siRNA or microRNA. These molecules mostly attach to 3' UTR of the mRNA molecule that they control. So it is extremely important to know exactly how big is the transcript, and it is 3' UTR region. So if we know this information, we can understand these mechanisms, how they, for example, one transcript could be silenced with siRNA or miRNA. So these are this is another benefit of 3' RNA sequencing. And here I will present you the experimental design that we use. We basically took the adipose tissue from chickens. We isolated RNA samples, and then we had overall 61 samples. 20 samples was used in for both RNA-seq and 3' RNA-seq, but 41 samples we had only used for 3' RNA sequencing. So after sequencing, so in this case we have 20 samples which has both RNA-seq and 3' RNA-seq. This will allow us to compare these two technologies to see if we can achieve high accuracy with 3' RNA sequencing. So we did as typically we do in lab. After RNA sequencing, we check the quality of RNA using a bioanalyzer to make sure that the RNA has high quality. We use RNAs that has 8 or higher than 8 integrity value. So after quality check, we followed the 3' RNA-seq library preparation step. So this is the protocol. You can find more detail about the protocols in Mold et al. Published in Nature Methods. So I wouldn't go through details of the protocol, but I would like to just emphasize here this part, first step, that the primers that are used to amplify or synthesize cDNA, they have a complementary T which attach to poly A tail of mRNA. So you have here poly A tail, and the primer would have poly T tail, so it would be complementary to poly A tail of the mRNA. So we will back to this point again because this is an important thing to consider in our analysis. We will see where this is important. So after synthesizing cDNA library, they are sequenced, and also these are more information about how sequencing can be done. You can find from this paper in Moll et al. So I will go through my talk will be how we analyze this data after sequencing. So first we check the quality of libraries. This is before sequencing, actually, which is also important point to consider because the amount of cDNA is much lower than what you get from conventional RNA-seq method. So you need to use high sensitivity DNA assay. So these are, if you use a typical DNA assay, it would not be detected, but you should use high sensitivity DNA assay. And we check all the cDNA libraries that we generated to make sure that they have high quality, and we see the product in product. When you do this DNA assay, you're going to see the product which is indicating there's enough amount of material for sequencing. And then we followed this protocol for DNA data processing. So we took 61 samples from abdominal fat. We did local alignment mode using Bowtie 2. And with this, we got 90% of the reads mapped to the chicken reference genome, which is total number of reads will be 123 million reads. After this, we went to the second step, was quality control. In quality control, we looked at, we excluded all these low quality mapping, the reads that had more low quality. For example, the criteria that used was less than 10. And after that, we excluded repetitive sequence or sequence with poly A stretch. I will come back to this point later on. So this was one of the criteria we used to exclude all these repetitive sequence and poly A stretch. And after we end up with 36% of the reads, which was 49 million reads. And in next step, we detected the position of 3' UTR. This basically one of the advantage of the technology that I mentioned. It allows to detect 3' UTR. And then we identified alternative polyadenylation, and I will talk about this later. And final step was using HT-seq. HT-seq is a software for quantification of gene expression. We use HT-seq to quantify amount of transcript from each gene and in each sample. And then because as I mentioned, we had both RNA-seq and 3' RNA-seq data, we use these two data sets to compare whether we have high accuracy. Accuracy is comparable between RNA-seq and 3' RNA-seq. So we had, for this analysis, we had 10 high and 10 low feed efficiency chickens. This has been studied before, a paper published, and that we compared high feed efficiency with low feed efficiency chickens. So we use the same data set to also do the same thing with 3' RNA sequencing, and to find out how high is the correlation between these two technologies. And I'm now going to some of the challenges that we faced when we were trying to analyze data, process data. So one of the challenges is basically what you see here. We have an intron, and this is an exon here, and this is the part with a solid box, but thinner than the other solid box. It is 3' UTR region. So this is gene model in current gene annotation file showing that the gene model ends in this point here, but when we mapped our 3' RNA-seq data, this track shows 3' RNA-seq data, and then we mapped these 3' RNA-seq data. These are our sequence reads from 3' RNA-seq. All of them, almost all of them map to this region. So we see that this region is much farther than where transcript ends. Transcript ends, you can see the transcript ends much shorter, but obviously this gene model shouldn't be correct and should be extended to cover larger region. So to fix this problem, we decided to basically we use a parameter to extend all genes up to 5 kb, and if we find a we found in location within this 5 kb that our reads map to, we consider that as an legitimate 3' end of the gene. So we will have more examples in following slides. So with this parameter, we improved some gene models, but still we had some other issues. So this is an opposite example. So I forgot to mention in previous slide, you could also see RNA-seq because we had both 3' RNA-seq and RNA-seq data. In RNA-seq, this is consistent with, we can see that RNA-seq data also shows that there is lots of reads mapping to this area, but there's nothing in gene model showing that whether there's a gene or there is not a gene. It shows that there is no gene here, but obviously this reads, all RNA-seq reads belongs to the genes that was ended here. So this gene needs to be extended further because both RNA-seq and 3' RNA-seq shows that there are expression in this area. So going back to the continuing with the example that is opposite to the example that I showed. So here we have a 3' RNA 3' UTR that ends here. In this case, gene direction is from here, and gene direction is from right to left. It is in minus strand. So we don't see any sequencing reads mapping to the end of the transcript because our 3' RNA-seq data should map to the end of the transcript, but we don't see any reads mapping to the end of transcript. But we see most of the reads, our RNA-seq data map to this region. And a little bit of them, like a few sequence map here, but most of them maps to the previous, as I indicated here, this region. So obviously this gene model would also be wrong. It needs to be shortened. It is opposite of what we just talked about it. This gene is in database, it is longer than it's supposed to be. So we also fixed this problem by just basically finding out where is the end of the transcript and cutting that and reducing to the smaller transcript. So this is another problem. When we were looking to gene model, you can see there's an intron here, there's an exon here, there's another intron here. And in our 3' RNA-seq data, we found that some reads, like you can see here, these reads are mapping to exons, and also these reads are mapping to exons. This exon is within the gene, so it is unexpected to see this. These reads are mapping to exon within an exon in the middle of gene. So we were curious about what is going on here. So what we did, we just zoomed in to see what happened. Like you can see two regions here, and what we found out after zooming in, we saw the stretch of poly A. You can see here we have A and also 4A here and 2A's here, here, and there's more A's here, which basically comes from the fact that the technology, because as you remember from my previous slides, when we try to amplify mRNA, we use primer with poly A tail, which attach to poly A tail of the mRNA. But that poly A tail is added after transcription. But it is not poly A tail that are within the gene. But here we have a stretch which is within the gene. It is coming from DNA. It is not the poly A tail that was added after transcription. So whenever we have this situation, the genes, the transcript downstream of that and upstream of that will be basically end up in our cDNA library. So we have to come up with a parameter to delete all these reads because these are not real end of the transcript. The reason that we see the reads here because upstream of this read, there is poly A stretch. So to fix this problem, what we did, we looked at all these transcripts. For example, when we found a location that our RNA 3' RNA-seq data map, in this location, we consider the endpoint here. From endpoint, we come -10 toward left and +18 towards right. And in this region, -10 +18, which is 28 base pair, we looked to find if there is 12 A's in a sliding windows of 18 base pair. So this was one of the criteria, 12 A's in sliding windows of 18 base pair, or we were looking to see if there's 8 consecutive A's. So we come up with these parameters after fine-tuning, after trying different things, and also by consulting with Lexogen. Lexogen is the company that is providing these kits, and they had more experience on how to handle these data. And after consulting with the company and also fine-tuning, we come up with this criteria. So applying these criteria, we basically eliminated lots of reads that was mapping to this kind of region. So we had also another issues, like you can see here. We have in left side we have gene A, which is in positive strand, and in right side we have other gene B in minus strand. So in gene A, the direction that is going is from left to right, and gene B going from right to left. So these are because they are in opposite strand. So the question was, because you can see all these reads between gene A and gene B, and it was not clear where these reads belong to. Are they belong to all these reads? This is RNA-seq, based on RNA-seq. So whether all these reads in RNA-seq map to gene A, or they belong to gene A, or they are belong to gene B. So based on RNA-seq data, it is not clear where they should belong to. But we use the 3' RNA-seq data to figure out where they belong to. You can see here we have 3' RNA-seq data, and it is not very clear in this slide, but these reads have direction. It shows that reads are going to this direction. So that's obviously all the transcript that is mapping here belong to gene B, but gene B starts here. But this in gene model, this region belongs to gene A. So if we use RNA-seq, we're going to count all these transcripts to gene A, but actually these reads belong to gene B. So we can separate these two genes here. Gene B will be from here, but because in here we have low expression of gene A, we cannot really determine with high confidence where gene A starts. We can just say gene B starts here, but based on the data that we have from adipose tissue, this gene doesn't express much in adipose tissue. So we need to look at another tissue to see where this gene expressed higher. So we're going to look at another example that both genes are expressing higher. For example, in this case, we have the same scenario, but both genes are expressing higher. We have gene A in plus strand and gene B in minus strand, and we also have all these reads in this area. And we don't because the transcript for gene B ends here, transcript for gene A ends here. This is based on current gene model annotation. So we don't. This doesn't really because we see all these reads between two genes based on RNA-seq. We can't tell where they belong to. So we do after using 3' RNA-seq. We have 3' RNA-seq here. So it shows that all these reads belongs to gene A, not gene B, because the reads are this. You can see that the sequence reads from RNA-seq, 3' RNA-seq is mapping here, and we can determine where each gene starts. So obviously the model gene B has the correct model because transcript starts from our reads are mapping to the point that this transcript ends. So we can basically be confident that gene B has correct model, but gene A has the gene model is for gene A is not correct. Another thing that we can pick up from this picture, we can see there's multiple sites that our 3' RNA-seq data is mapping, and this is where what I mentioned in the previous slide, that some genes have multiple cleavage sites and there's multiple polyadenylation. So using 3' RNA-seq, we can also determine this multiple polyadenylation sites. So this is just the same slide but zoomed in. You can see that the reads has direction. This reads going to from right to left. This reads going from left to right. So we can say that which genes they are belong to. So this is another case for one of the problems that we saw is based on gene model. You can see these are two exons and this is an intron, but we saw in both in RNA-seq and 3' RNA-seq there are lots of reads mapping to intron. So obviously this would be not, this part would not be an intron. This looks like a 3' UTR just started after this exon here, right here. So all the transcript after this exon should be like an extension of 3' UTR region of this exon. So this is another case that we fixed it with parameters that used for data analysis. And this is also interesting things that you can see how many alternative sites for polyadenylation exist in the transcript. And we are looking to 3' RNA-seq in 61 samples pooled together, and also 3' RNA-seq, but this time looking one single sample. And on here we have RNA-seq data. So based on this graph, we can see even in one single sample there are multiple sites of polyadenylation. Part of it can be because of alternative splicing, and part of it could be because of usage of the 3' UTR. So this with 3' UTR, we could also figure out how the alternative polyadenylation sites in the transcripts in the genes. So after doing all these improvement and filtering the data, cleaning the, for example, looking poly A stretch and extending 3' UTR, we were wondering how good we were doing. So we looked at these are, as you remember from earlier starts, there's polyadenylation signals. These are typical non-polyadenylation signals, which is called consensus sequence. So this, as you can, like the first one is the most frequent one in mammalians and also in birds, as we can see here. In chickens also, this is the most abundant polyadenylation signal. Based on current annotations, which is showing by blue, the color is blue for the current gene model. The color is red for our improved annotations. So our improved annotation, we finding 2 times more of these signals in the transcript. So it shows that the improvement we did, it actually improved the gene model annotation, because you can see that it's almost more than double the number of polyadenylation signal. And the second one also is doubled. So this is the second non-polyadenylation signal. So we can see that there's also 2-time increase in this signal in our transcript. And overall, if we put all together, here we have percentage of transcript with at least 1 consensus sequence. In current gene model annotation, there's only 64.25% of transcript with the non-polyadenylation signal. But when we use our improved annotation, we found that 91.8% of the transcript had at least 1 consensus sequence. And if you go to this category, percentage of transcript with 2 most abundant, most common polyadenylation signals, which is AAUAAA and AUUAAA. So based on current gene model annotation, there's 39.42%, and based on our improved model, it's 80.76%. So this clearly shows that using our improved annotation, gene annotation, it could help to identify more of these polyadenylation sites. And the things that we did, the criteria that we use, it helps to improve the gene model. So in this part of my presentation, I will actually talk about the differential expression analysis, because we had both RNA-seq and 3' RNA-seq data. We can compare RNA-seq with 3' RNA-seq. We had, from RNA-seq, we found out 217 significant DE genes, differentially expressed genes, between 10 high P deficiency and 10 low P deficiency broilers. These are meat-type chickens. So total number of reads from RNA-seq, we had 60 million read per sample, which was done based on four samples per lane. And in 3' RNA-seq data, we had 2 million per sample. You can see 60 million versus 2 million. So there's big difference between number of reads per sample. So this was done, all 61 samples were pulled and sequenced on 1 lane. So the results was really shows high improvement when we use improved annotation. So in current model, the correlation is 0.782, correlation between RNA-seq, log2 fold change, and log2 fold change 3' RNA-seq. So we had correlation. This is correlation between 2 technologies. So correlation 0.78 compared to correlation 0.91, which shows all improvement that we did for gene model using 3' RNA-seq data, helped us to reach to high accuracy. But even with this 0.916, we think there's room for improvement, because there are other problems that, for example, here we haven't still fixed this problem. We are working on fixing these problems. We have here gene model ends here, but there are 7 exons missing from gene model. So this is indicated both based on RNA-seq and 3' RNA-seq. And if you zoom in, we're going to see that our 3' RNA-seq data mapping to here. So this gene basically need to be extended farther, but we haven't extended this gene. If we fix this type of problem, we may still improve our correlation. Another problem was, as you remember, we also used the criteria to remove repetitive sequence, and that sometimes, because end of the transcript, there are repetitive sequencing at the end of the transcript. When we use this is like before QC and this is after QC. Before QC means we didn't remove repetitive sequence. After QC, we removed the repetitive sequence. So once we did QC, and then all the reads that were mapped before to this location, this is 3' RNA-seq read. So all these reads are eliminated. So this is one of the problems that we are trying to fix when we have repetitive sequence at the end of the transcript. Another example, there is also quite, I think, how I am doing in terms of time, how much time I left. Moderator?
You're fine to go on as long as you wish. You should finish what you need to say.
Okay, thank you. So yeah, there's like problem we can see here. There is a gene which basically it is within another. This gene ends here, but based on our RNA-seq and 3' RNA-seq data, the gene should be extended up to here. And we have another gene which I actually showed here. It is within this gene. So we haven't still fixed this problem, how we address this type of problem. And another problem, okay, as you remember, I mentioned the extended genes up to 5 kb, but sometimes we find reads mapping to even farther than 5 kb. We are still trying to solve this problem. So when some of the reads mapping to even farther than 5 kb. So at the end, I would like to just conclude, give a few conclusions. As you see, we haven't still finished all the way down, but I feel that we are confident to make some conclusion from this study. We have, first of all, it's going to be big cost saving. If the goal is just to quantify gene expression, we can make some cost saving here, because this method is require like very low amount of sequencing. So there's going to be cost saving, and this helps you to scale up sequencing projects to use that sequencing for population studies. And also the 3' RNA-seq, as I mentioned, it allows for accurate detection of the 3' end of the transcript, which enables verification of the current gene model annotation. Based on this data, we re-annotated 8,157 genes out of 10,693 genes. These are 10,000 genes is the number of genes that express in adipose tissue based on our data. So basically, we could we re-annotated 76% of the gene, and we were able to characterize alternative polyadenylation sites. And that means we had found that 7,354 genes had alternative polyadenylation sites, which is, if you consider 10,693 genes that are expressed in adipose tissue, this will be almost 70% of the genes. So 70% of the genes shows alternative polyadenylation, which is huge amount of variation. People would be interested in looking association between variation in polyadenylation and phenotype. So because sometimes transcript with different lengths, they do different job, or they have different time long life, lifelong. So it would be interesting. People study this type of polyadenylation using the technology that is not very expensive. So at the end, I would like to thank people who actually helped me to do this project. Basically, we had most of the bioinformatic analysis is done by Vision tool, grad student in my lab. And Juju and Mustafa Özcan, they did a library preparation. So they basically did most of the work. So I would like to thank them for their collaboration. And also the financial support that University of Delaware provided. And also thanks the company Lexogen for the kit. And if there is any question at this point I would be happy to take.
Thank you, Dr. Abashed. At this point, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to get to as many questions as we can. And your first question is, where can I buy the kit? Is Lexogen the producer of this kit?
Yes, they produce this kit. Like we bought from Lexogen, so they have. I saw them in this year in PAG. They still continuing selling this kit.
How do you remove the internal priming events associated with the oligo-DT priming of reverse transcription?
Yeah, that was actually one of the issues. We couldn't eliminate at that point, but we do bioinformatically. As I actually explained, we use a criteria to eliminate reads, but that happens. Internal priming happens, which generate some problems, but we could remove them using bioinformatics.
What is the role of the poly A tail at the 3' end of mRNA?
Yeah, that's a good question. They basically poly A tail has several roles. One of them is stabilizing RNA. So a poly A tail, if it is shorter, DNA have will live shorter. But if it is longer, RNA will live longer. So there are multiple roles for poly A tail in the end of the transcript. The most important one is how soon RNA will be degraded depends on the length of the poly A tail.
Thank you. Can we detect alternative splicing using this method?
Yes, I actually talked a little bit. So yeah, you can do it because this targets the end of the transcript. If you have one gene with multiple alternative polyadenylation sites, you could be able to find reads mapping to that site.
How many reads per sample are, in your opinion, appropriate or sufficient for a quant-seq analysis?
So we used, in this study, we had 2 million, which provided decent results. So I think 2 million, but I would also recommend to check with the company to say if they have another recommendation. Based on our results, 2 million reads per sample provides satisfactory results. If you compare RNA-seq with 3' RNA-seq, it gives you 0.91 correlation, which is we think that it is a good correlation between these independent technologies.
What read length has been used for sequencing?
We used 100 base pair, single end, but my recommendation to going forward will be paired end, same size of the read length, so it's 100 base pair, but paired end reads will be better than single end reads because sometimes, one of the problems that I mentioned, repetitive sequence. So if one of the pairs map to repetitive sequence, you can also use another pair. So it could probably reduce those problems. So we plan to use paired end sequencing later on. This was single end, but future project we would like to use paired end. But 100 base pair in terms of length of each read, it would be good enough.
Do you isolate polyadenylated RNAs before preparing the libraries using an oligo-DT? Sorry, I mispronounced that.
No, it is basically it doesn't have that stage. Because you're using primers that amplify that targets poly A tail. So this protocol, if I'm not wrong, this protocol doesn't have this mRNA purification stage. It's just use of total RNA to start with.
What is the mapping quality?
Mapping quality was good. We lost 10% of the reads after QC for mapping quality. So this is like even higher than what you can get with RNA-seq. So based on our data, mapping quality was good.
Why do you remove reads with poly-A stretch? Wouldn't trimming be better?
Because those trimming wouldn't solve, because those reads need to be eliminated, because poly-A stretch is not representing the end of the transcript. They are within the transcript. So if your goal is to detect polyadenation, it will give you false positive results, because you're going to see reads are mapping to the location with poly-A stretch, which is falsely identified as a polyadenation site. This would be a problem for detecting polyadenation site, but if the goal is to quantify gene expression, you don't need to remove those reads, because if you have two groups, let's say treatment group versus control group, and both of them going to have the same, both of them going to have reads mapping to the region with poly-A stretch. So when you compare two genes, one gene in two groups, both groups going to be roughly equally affected. So for now, answer will be you need to remove those poly-A stretch when the goal is to identify polyadenation, but you don't need to remove them when the goal is just to quantify gene expression.
Thank you. While extending the UTR, how do you handle the fact that there may be other genes in the 5 kb window?
That's a good question. So we put another criteria that when that 5K extension hits another gene, so stop that there. Stop before moving inside that other gene. So we extend it as long as there's no other genes in the downstream.
When you find, this word wrong is in quotes, when you find wrong 3' ends upstream or downstream, the annotated 3' end, isn't it possible that it is actually alternative poly-A sites specific to your cell type or to the condition tested, allowing the existence of the, in quotes, wrong annotated 3' end in another condition?
And what was the question number? Can you please?
12.
12.
Can you see that okay, or would you want me to read it again?
Yes. Yeah, I see that.
Okay, good.
Give me a few seconds. Just I need to see.
Yeah,
full.
It's a long question. Take your time.
I don't see end of the questions like in here.
Oh, okay. So look below. Look. So you'll see the top if you highlight number 12. Look below near the end of the page.
Okay.
The whole question.
Okay, I see. Yes, that's a very good question. Yes, it could be that gene model is based on one specific tissue or cell type, but we found another site which comes from the tissue that we use. So that is really a good question. That's why we have to have multiple tissues. If we want to create a gene model, need to be multiple tissues. So if I'm working in one tissue, so I would be confident that the gene model also cover where my gene ends. But yes, that's possible. This technology needs to be applied across more tissues to be able to provide the models that will work with every tissue.
What is the frequency of transcripts in which you don't have 3' UTR sequence compared to RNA-seq?
What is the frequency of the transcript in which they don't have 3' UTR sequence?
UTR sequence compared to RNA-seq. You want to take that one later?
Yes. Let me 3'. All these transcript, mRNA transcript, they need to have 3' UTR sequence. So we haven't looked at this to come up with a good number. So we haven't done this analysis yet.
Okay. Any idea how many genes, if any, have the same 3' sequence and can therefore cannot be distinguished from the data?
Same 3' sequence. We didn't find this type of maybe in some species that had better gene annotations, for example, humans, and there will be many genes with the same 3' sequence. But in chickens, we don't have that quality of the annotation file, so we don't see many genes utilizing same 3' UTR.
How much do you clip off the reads before aligning?
We don't clip any of the reads because we put the quality parameter. If the quality is lower, the reads won't map. So we didn't clip anything.
Okay. How much total RNA do you use to begin with?
Total RNA, that I don't remember, but they are indicated in the protocol how much we follow how much the RNA was recommended based on the protocol.
Dr. Abash, do you have time to answer a few more questions? Do you have
Yes, I do.
Okay. So the next one is, where can I find a full description of the methods used to correct gene models and overall corrections for the issues with 3' RNA-seq described in the seminar?
We haven't still published data. I would be happy to help if there is any question, if someone wants to know about details. But it's still, this study we haven't published yet. There is no information published available.
Okay. We have someone else who's just thanked you. Several other people have also just written in thanks for your presentation. Here's another one. How about alternative splicing transcripts that skipped some middle exon?
Because we are looking to the end of the transcript, so we wouldn't detect the old those that are skipped the middle exon. So we will see that the transcript there is more. So that would require other things because we don't really looking for all that is alternative transcripts that is generated based on splicing.
What instrument is used for the gene expression analysis and for analytical expense, please?
Gene expression analysis, HT-seq, the software is called HT-seq. If the question's asking the method of quantification, it will be HT-seq.
Okay. And I may have missed this as I joined late. When you make the libraries, how do you check their quality before proceeding to sequencing?
Yes, we use a bioanalyzer. Also I emphasize that we use high sensitivity kit because the amount of cDNA library is lower. So you have to use a high sensitivity kit. So the answer will be bioanalyzer using high sensitivity kit.
Is there any limitation?
Could you please read also question number? I can follow which question
yes.
was.
Oh, of course. The next one is 22. And the question is there any limitation to using fresh samples versus fixed tissue samples? How much of mRNA extraction amount is needed?
We haven't tried with fixed sample. We use fresh sample all the time. So I would refer to this question to be asked from the company. So how much mRNA extraction will be started with minimum amount of RNA extraction that we had was 3 nanogram per microliter. But I think but I don't exactly remember how much we put. We use the recommended amount that's recommended by the protocol.
Okay, now we're on 23. Can you please elaborate on the methodological usage of 3' RNA sequencing with RNA silencing?
So because this is just when in RNA silencing, those small RNA like siRNA, microRNA, they attach to most of them attach to 3' UTR. So this is going to kind of when you find some transcript has larger 3' UTR compared to shorter one, and if you have a microRNA that attached to 3' UTR region, so it wouldn't have any impact on the transcript with shorter. If the mRNA mapping to the region that is not present in your transcript, so you shouldn't be worried about the impact of that microRNA on your transcript. So this is kind of like you couldn't do any experiment to validate it, but you can hypothesize that. For example, if I have a transcript that is shorter, that has shorter 3' UTR, and there is no site for microRNA to attach to that transcript, so there's less likelihood for the transcript to be impacted with microRNA.
Okay, now we're on 24. How can a 3' RNA-seq approach be helpful in polyploidy models?
Polyploidy. I have to think. I don't know. It is not right now obvious to me how it could be helpful, because you're going to have in polyploidy, supposedly there's going to be more of the transcript from same gene. So if there is a variation on gene expression that arises from polyploidy, for example, one chromosome produce transcript which is different from other chromosome producing the same transcript. So if there is a variation at chromosome levels, it could be helpful to pinpoint, for example, in your system, if you have tetraploidy and genes will have 4 copies of the gene, and each copy may utilize different alternative polyadenylation sites. So it could help, but we never tried to see how it could be, because we just look at the system that was deployed.
Number 25 is, how can we select the genes?
Not sure what the question means, how we select the gene, because we don't select any genes. We are basically looking all the genes, because we are studying like RNA-seq. In RNA-seq, you sequence all the mRNA in the sample. The same thing here. You're basically sequencing 3' UTR of all the transcript in your sample.
Number 26 is, what was the significance of the complementary poly-T tail?
Because we talked about we use primer that has poly-T to attach to poly-A. So this also can attach to the region of the genome that has poly-A stretch and produce false positive. So that's why we wanted to remove those sites to not have false positive. So if the question is talking about the issues that cause with poly-A stretch, so only the way that we can solve it is using a bioinformatics way.
Is 3' RNA sequencing applicable with cytoplasmic intron retention mRNAs?
Okay.
That's number 27.
I'm not very familiar with the cytoplasmic intron retention by mRNA. It sounds to me, based on this question, it looks like in cytoplasmic mRNA, sometimes RNA intron is retained as an exon. but I don't think that would be helpful because if intron is retained within the gene, you couldn't be able to detect the 3' RNA-seq. But if that, yes, I think it is not going to be helpful if intron retain, because the last things that we have is exons. So this technology targeting the exon, last exon. So if you have an intron retain, it wouldn't be detected based on 3' RNA sequencing.
Thank you. I would once again like to thank Dr. Behnam Abasht for his presentation. Sorry that I mispronounced your name, Abasht, for his presentation. Do you have any final comments for us?
Not really. I would like to thank everyone that listened to this presentation. If there is any questions and if I can help, I would be happy. Just send me emails. I have my, you can find my contact information from University of Delaware's website. So if there is any question that I can help, please send me email.
Great, thank you. I would also like to thank our sponsor, Lexogen, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through September 15th, 2016. You'll receive an email from lab groups alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.